Surely you recall the second Koch postulate, the requirement to grow suspect pathogens as pure cultures. But how can you satisfy such a condition for a total parasite? Viruses are completely dependent on the cells of their host for replication. When viruses infect bacteria, bacteriophages, the pure culture condition is feasible. This is because the host organism itself, to start with, can be grown in pure culture. Thus, phages infect bacteria that are in essence a pure culture of single cells. Then the viruses replicate within them to be released into the culture broth where they can encounter and infect more bacteria. The situation, however, is far more complex when one wants to study viruses that infect animals made of different kinds of cells that form various organs, such as the heart, lungs, and liver. Trying to decipher the details of viral infection by examining a living being, a whole mouse or person, for example, is literally impossible. If only one could culture pure animal cells of specific organs and use these just like it's done with pure bacterial cultures, then we could systematically study the biology of viruses that cause diseases in humans. Indeed, this became possible with the invention of cell cultures, a technological breakthrough that has revolutionized the field of virology and has since allowed us to discover how viruses work. Today, there are a number of ways to culture cells in the laboratory. All the methods entail growing cells in sterile plastic petri dishes or special culture flasks. These contain growth medium rich in nutrients and various growth factors and are all incubated at 37 degrees. Most commonly used are cells that have been adapted to these special growth conditions for many years and are by now well-established cell lines. Cell lines can be derived from plants, insects, mice, monkeys, and even humans. Certainly the most famous immortal cell line is HeLa cells. These are cells that were derived from the cervical cancer taken from Henrietta Lacks just before she died way back in 1951. HeLa cells were subsequently cloned in 1955 and have since been grown continuously in scientific laboratories all around the world. Very often the cells in culture grow to form continuous single layers of cells. These stick to the bottom of the culture dish and form a monolayer. When cells in monolayers get so crowded that each cell touches its closest neighbor, they stop dividing, a process called contact inhibition. Interestingly, cancer cells are not contact inhibited and grow out of control to pile up and form heaps of cells, much in the same way as cancer cells form tumors in our body. So you see, we can learn much about what goes on in cells from the way they grow in culture and the shapes they form. When cells in culture are infected by viruses, the viruses can affect how these cells continue to grow, spread in culture, form heaps of transformed cells, or die creating plaques. These aberrant growth patterns are referred to as virus-induced cytopathic effects, CPEs. One curious CPE is the merging of infected cells. The cell membranes fuse to form one giant cell with multiple nuclei. These multi-nucleated cells are called syncytia. Virus can also cause cells to lose contact inhibition and lead to the formation of transformation foci. Clearly, the replication of viruses in cell culture has been enormously important for science. It has also allowed the mass production of large amounts, literally grams and kilograms of pure viruses that can be harvested for the commercial production of vaccines, as we'll see later on in the course. In the coming unit, we will learn about how viruses select their hosts, replicate their genes, and express their proteins to ultimately assemble new viruses and thus complete the replicative cycle.